So now that we know the role that each of these hormones play within metabolism, let's look at how they elicit those responses. So generally speaking, we can categorize cell signals as either binding to membrane-bound receptors, such as the insulin receptor here, or binding to an intracellular receptor, such as glucocorticoids or cortisol. Insulin, glucagon, and epinephrine all bound membrane-bound receptors, and insulin binds a receptor tyrosine kinase, whereas epinephrine and glucagon primarily bind G protein-coupled receptors. So we'll focus on those here. For the majority of the course, with respect to insulin, glucagon, and epinephrine, we'll be focusing on these cytosolic responses, which change the phosphorylation state of metabolic proteins. The change in phosphorylation, we'll see, will influence the activity of the enzyme. So if we look here at insulin signaling, it starts with binding of our ligand insulin to its receptor here, which is a dimer. Upon ligand binding, the dimerization causes an autophosphorylation event of these exposed tyrosine residues, denoted here by Y. These phosphorylated tyrosines serve as a docking station for the adapter protein, the insulin receptor substrate. This interaction is both through phosphotyrosine domains as well as through plextrin homology domains. So following this docking event, you'll notice several other tyrosines on the insulin receptor substrate, which can then be subsequently phosphorylated to serve as additional docking stations for the recruitment of additional proteins, one being GRUB2 and the other being IP3 kinase, which is phosphatidylinositol 3 kinase. This can then activate the downstream kinase, PDK1, which is phosphoinositide-dependent kinase. The activation of GRUB2, on the other hand, will activate a MAP kinase cascade. These downstream mediators here just give you the breadth and scope of the impact of insulin signaling. What's important to note are these proteins above, which we've just discussed. It's also important to note that this signaling cascade can be terminated by the presence of the protein P10. But through this signal amplification is how insulin then elicits the multiplicity of cellular response, including protein translation, enzyme activity, gene transcription, and cell growth. So epinephrine and glucagon primarily signal through G protein coupled receptors. And what you'll see are these receptors are characteristic of these 7 transmembrane domains. They're also associated with, although not bound to, this other complex of an alpha subunit, a beta subunit, and a gamma subunit. There's typically also a downstream enzyme effector. So what you'll see is that when hormone is bound, you have a conformational change in these transmembrane domains that changes the plasma membrane in such a way that facilitates first the exchange of GDP for GTP and then the release of this alpha subunit. This will then activate a downstream enzyme. In the case of the G-alpha S subunit, the downstream enzyme activated is adenyl cyclase. We'll also look at the activation of phospholipase C by a GQ subunit. So for a G-alpha S subunit, the downstream enzyme activated after GTP is bound is adenyl cyclase. This will then increase synthesis of cyclic AMP. Cyclic AMP will ultimately activate protein kinase A, which will elicit a response that leads to phosphorylation of lots of proteins and a modification of a physiological effect. Cyclic AMP is degraded by phosphodiesterase and thus serves as a means of terminating the signal. It's also important to note that cyclic AMP can also serve to regulate transcription, and it does this by binding the protein CREB, or cyclic AMP response element binding protein. This protein can then bind the DNA enhancer element, CRE, and modulate transcription of downstream genes. In contrast to G-alpha S subunits, where cyclic AMP is our second messenger, GQ subunits, which are characteristic of some epinephrine signaling, will activate the enzyme phospholipase C. Phospholipase C is responsible for cleaving PIP2 into inositol triphosphate, or IP3, and diacylglycerol. These two second messengers will serve to do two separate things. Diacylglycerol will activate protein kinase C, whereas IP3 will enhance calcium release from the endoplasmic reticulum. In both cases, you have activation of a kinase, which will then lead to the phosphorylation of downstream proteins. This slide summarizes the reaction catalyzed by phospholipase C, and here you can see the molecule bound in the membrane PIP2 being cleaved into our diacylglycerol molecule and inositol 1,4,5 triphosphate. So that summarizes the major signaling cascades by insulin, glucagon, and epinephrine. So let's finish with a question. So a novel signaling cascade is discovered that you suspect activates a G-alpha S subunit. So which of the downstream events would be consistent with this discovery? So downstream of G-alpha S, if you remember, we have cyclic AMP. And cyclic AMP will be increased when G-alpha S is active. So that eliminates choice B. We also know that phosphodiesterase decreases cyclic AMP through degradation. So we can eliminate choice C. So cyclic AMP is synthesized by adenyl cyclase. So it's also not likely that adenyl cyclase is inhibited, leaving us with choice A, which is the correct answer. Downstream of an activated G-alpha S, you'll have activation of adenyl cyclase and increased production of cyclic AMP.